G'day, how are yous? Hope you're all well. This is another study session for Environmental Biotechnology, a university course that I took actually last semester, and I didn't have an exam slot, so I ended up sitting the exam this semester today in 1 hour 41 minutes. So I'm going to go through some questions, and then if there's enough time at the end, I'll share some of the slides from the class. And it's a pretty interesting one. It's to do with microbes, all to do with microbes. So it's fascinating to understand the microbes, how they interact with each other and what goes on, and etc. etc. All right, let's start doing it. Share screen. So for the people, so I'm not really going to be doing the questions and stuff very much. They'll be at the end possibly, but it's just more for the slides. So share screen, so I'll start now. Alrighty, so back to here. So this is the right one. Week 3, was it week 3? Week 2. I'll stop sharing for a sec while I set up. Sorry about that. There it is, week 1. It's in the wrong way. So there's 2, 1. There it is there. Okay, no worries. All right, get back to you and press the share. Not invite. Share. Entire screen. All right, here we go again. This should be week 1. There it is, week 1. So, with respect to the evolution of biotechnology, which of the following options represents modern technology? So, it's for plant breeding. No, it's recombination DNA and DNA. So it's D. Majority of biotechnology processes relevant to environmental applications are microorganisms based because you can read through them. C, relatively inexpensive. Identify the primary crisis that is challenging environmental science and biotechnology: climate, water, population density, yet population density. Principal aim of the field environmental biotechnologist is to produce, no, to minimize, yes. Control, yes. Minimize, B. Is this a true or false? Eukaryotics remain the dominant drivers of the biogeochemical cycle despite strong anthropogenic influences. No, it's prokaryotics, so it's false. Identify the correct organism biosystem hierarchy in the ecosystem. So it starts with genes, not organs. Genes are required to make cells to make organs. B. Interactive space working for ecosystem functions include chemical landscapes, catabolics, anabolic, antibiotic, sorry. I'm really bad at English. Well, the abiotics is the everything but the microbes, so it's the A. Everything but life. Characteristics of an ecosystem is a result phenomenon of all interactions include biotic components' ability to respond to abiotic conditions. Is it true or false? It's got to be true. Choose the correct statement of the food chain. Alright, the numbers, secondary carbon ores will be less in number than primary. Yeah, that's how it works. If we study any general, yep. Food chains, longer 5 levels. Energy transfers to tropic. Oh, no, that can, so it's B. C? No, it's B. Major difference between energy flow and nutrient cycling is? After the death of the protoplasm, nutrients released and reused. Yes. So B is good. Nutrient transfers from one level to the other. 5%. No, you can't. It's not 5%. Both they're correct. No. I'll go with B. B. Microbial ecology explores diversity, effects, and the abundance of microorganisms. D. state whether the following is true or false. Microorganisms are presently in a habitat where microhabitats are not suitable, so it's true. Many microbes establish relationships with other microbes. When species A is in a relation with species B, the species B is not believed, not harmed. The relationship between A and B can be termed as, well, it's a co-beneficial. So it's co-mutual A. Yep. Which among the following has the maximum effect of the types of microbe activities that could be possible in an ecosystem? Which has the maximum effect? That's what's going on there. The symbiotic food web, nutrient cycling, possibly micro species. Well, yeah, all the species, if they're not there, they're not blooming there, mate. Good. Which of the following techniques are used in ascertaining if two strains of bacteria belong to the same species? A total number of species present in a microbial colony indicates its abundance. A. In an undistributed organic role, rich soil, species richness and species abundance will be pretty good. Species richness will be high. Yeah, it's a given. Species abundance will be quite high too. I reckon it's moderate. True or false? Prime or realized niche of an organism is usually smaller than its fundamental niche. Yes, it is. The term biogeochemistry was coined by Vladimir Banansky. Hello to everybody in Russia. No, actually, Russia's bad. The government's bad. I shouldn't have said that. Jesus. Sorry about that. Choose the correct statement about biogeochemical cycling. Biogeochemicals are linked. Yeah, they are. Energy and biochemicals are the type from light only. No. That means it's a autotroph. C. D is the answer. D. Thank you. Superbugs, which are more efficient in degrading hydrocarbons or other pollutants, must have the following characteristics. Increased enzymes. Yes, enzymes is good. Expression of genes. Most pollutants. Expression of genes. It's B and A and B. D. Removal of phosphorus and nitrogen from waste can be possible by. No. Yes. Co-pyrolysis. Yeah, I want to destroy everything. So B. At-risk resources addressed directly by environmental biotechnology are, what's the water, and energy. D. Important system components that need to be identified by a modular to create a model for a particular environment are. It's got to be all of the above. They're all pretty important. Identify whether the correct statement below is true or false. Cultivation-dependent approaches refer to laboratory-based isolation of microbial strains. Cultivation-dependent approaches, they do. True. Identify whether the statement below is true or false again. Developing countries have potential to gain double benefits by investing in environmental, but yes, they do. Oh yeah, and eliminate hazards. It's so rad, biotechnology. It's pretty hard, mind you, but it's just fantastic. I love studying microbes. Identify the different energy carriers that can be used while microorganisms utilize organic matters. Well, that's organic's pretty, there you go, it's all of them. C, D, they're all of them up. Identify the most suitable tool for determining the substrate utilization by a particular microbial cell. Nanoscale. Nano is always rad. Next generation sequencing. If too much organic matter, biological oxygen demand, is oxygenized in the water, what will be the predicted outcome in the terms of the oxygen, the DO and the levels? Well, DOs will go down, 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 C. Toxic pollutants like tetrachloroethylene, ethane, can be removed from contaminated water by using which of the following organisms? Dihalococcodides species. Very good, very good. In omics approaches to study microbial colonies, which is the best studied to gain enzymes related to proteins? Which among the following is not practical goal of microbiology in an environmental engineering system? Developing bioreactors. Well, bioreactors already developed. You just put air in a mechanism to turn it. It's all to do with the turning mechanisms too. It's pretty full on, all the different types of aerations and systems you can build. It's pretty funky. Which of the following general aspects is needed to fulfill the practical goals of microbiology systems? All. Bacterial strain capable of utilizing inorganic compounds as a source of energy and CO2 as a source of carbon is referred to. Well, carbon source would be a autotroph, and lithotrophs is rocks, so you need, that's inorganics. Among these, which is true in relation to aerotolerant anaerobes? So they are aerobic. No, they're not. They have protective mechanisms against oxygen radicals. Yes, they do. Well, that's the answer. They die. No, that's they got the protective agents with the free radicals, and it's pretty cool. Like the SOD, PO. Anyway, a species spores produced by fungi, an asexual spore produced by fungi is a cornedia. Yep. Algae are photoautotrophs. Look at there. Yeah, hang on, I can explain it. Elemental composition of biomass can be in the formula of it's CH18, 1.8, O.5, and N.2. Average carbon content of microbial biomass is about 50%. Which one of the following is the options for this equation? We can see as it goes through here, it's going to give off, and that's going to reduce the hydrogen, the protons, so generate reducing equivalents used. I'll go with B. Metabolic and taxonomic diversity of microorganisms on our planet are the outcome of dynamic. B. Yeah, good. The cause of physiological diversity of microorganisms in environments could best be linked to selective pressures. Yes. D. Yeah, good. All forms of life need a source of carbon to make their living because it's not ATP. It helps in assembling building blocks. Yes, it does. B. And B. Chemosynthetic lithoautotrophs can obtain their energy from, well, chemosynthetic, that's the inorganic, and lithos, the inorganic, because that's the rocks. Autotroph is the sun, or they make their own energy. So it's inorganic, so it's all inorganic. B. Identify whether the statement below is true or false. In thermodynamically unstable resource that have provided selective pressures for microbial energy production strategies throughout evolution, it is the thermodynamically unstable resource. Yes, it is. In species reactions, transition from electron acceptors to electron donors depends on, well, it's definitely the redox and catalytic Mechanisms, yeah, because you've got to have acceptance of B, yeah, C and D, both A and B, C. If water samples are collected and analysed over time, the sequence of depleted electron acceptors will be, well, it starts from oxygen special electron acceptor, then it goes down from that. It goes to your nitrate, to your sulfate, and once it gets anaerobic, your carbon dioxide. A stimulatory reduction of CO2, NO3, and SO4 are part of, that's fermentation style stuff. That's when it's getting, well, there's no oxygen. See that? There's no oxygen, so it's got to be without oxygen. And it's a part of an anabolic cycle, because you've got to make it first to use it. So the anabolic makes it and catabolic uses it. So if they're reducing it, that's an anabolic, I reckon, B. Identify the statement's true or not. In absence of extracellular terminal electron acceptors, ATP production is hampered. In the absence of extracellular TAs, yeah, bloody oath it is. Because if we haven't got our terminal, like us for oxygen, if we haven't got a terminal electron acceptor, we're in strife. True. Identify the correct based on the statements. No, it's not 1. It's 2 and 3. B. 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 Identify the statement which is not correct. Well, that's right. It's static systems, because everything else is complex, as it's not stationary and static. Among the following statements, the majority of microalgae, which one would help us more identify the facts and points where perhaps it is not known properly? Maths always helps. See, microorganisms can be isolated from the environment system as per culture. Bacteria are generally subject to further analysis. These analyses include the term great plate count anomaly helps us understand there could be more number of bacteria present in an environment sample. Yeah. Yeah. Okay. Identify 0.1. Isn't that amazing? 0.01, that's mostly seawater, to 1% of mostly aerobic of total bacteria are culturable. This is true. Gene that is considered to be one of the best molecular markers for taxonomic. It's 16S rRNA. That's for microbes, because humans is 18S. Ribosomal nucleic acid. For taxonomic identification of most microbials present in the soil, most appropriate method will be done incubating test. Extract, yes, D. Extract. Metagenome, meta, do everything. Which is not the relation to the most probable plate number, most probable plate number, most probable. Oh, geez, I can't remember what it stands for. It gives appropriate message. It's not validate. Well, it's not. Yeah, most probable number. It's not to do with anaerobic, because you can't count. It's hard to do with that. Fermentation. It's B, I reckon. B, yeah. Identify the statements for dying, for the stain dying, cell dying. Which one's true? Which one is identified? Oh, the incorrect. B. Bacterial stain obtained from wastewater facility was found to be growing well under anox conditions without exterior electron acceptor. When provided with an organic carbon and nitrogen source, lactate and yeast, metabolic, so it was fermentating. Fermentative. Why do we need to adopt the culture and dependent methods for understanding microbial ecology? Because they're unculturable. B. They're not culturable. Genes which are useful in microbial analysis are C. Identified as true or amplified ribosomal DNA restriction analysis, one of the most common fingerprints. Yes, it is. Select the correct options for sequencing of a microbial community. So if I was going to sequence one, you want to extract everything. Yep, then you want to PCR it, and you want to sort it out in the library. You want to blast it through its blast library, and then separate the gel fluorosis. Use your PEG to separate it, polyethylene glycol, and then collect it and clone it. So yeah, A is pretty good. Extraction. I think this is good. Options, correct. Select correct options. So D's works as well. A and D. Add. Identify the correct technique to help us characterize the target gene of interest in a genetic soup. Quantitative polymerase chain reaction. B. What are alpha diversity parameters? Operational taxonomic units. Yep, your OTUs, species richness and abundance. C. Yeah. Ooh. Select one of the sequencing methods that can be sequence billions of nucleic acid fragments. That's a metagenome. Next sequence generation. The Sanger, that's the old school, that's the first one. Next generation, that's the fifth generation we're up to now. Sanger's number 1. Identify whether the statements are true or false. Transcriptomics is an analysis of your messenger RNA produced by a strain or community. It is. Yes, it is. Metagenetics can be determined by presence, taxonomic. It's all of them. Yeah, it's all of them. First generation sequencing of complex human genome was done by. Oh, this is the first one. Sanger. Yeah, Sanger. We all want a Sanger, remember? Which among the following is identified as major roadblocks? Definitely B. C is hard too. BC, rad. BC is so nice. I miss it a lot. Metatranscriptomics, living there. It's such a nice place in Canada. Can be used to determine instantaneous change in the community responsible for interpretations of gradients. True. Metagenome assembly is process of dividing sequence reads into continuous stretches of DNA called contigs. No, false. Bioremediation is managed as a spontaneous process in which biological acts on a catalyst, thereby remedying the pollutants. C. Something are most preferred candidates and major entities for bioremediation technologies. Which one do you reckon? Hint, it's to do with how fast cells divide. So it's B, because plants are twice as, they're like at a zero. Bacteria is sort of hours or minutes more so. Plants are up to 16 to 20 hours for cells to divide. Bacteria minutes. Fungi might be around that too, but bacteria are fastest. Microorganisms capable of organic pollutant bioremediation can utilize these toxic compounds. Yes. You want everything. D. Specialized bacterial culture with degenerative abilities are added into contaminated polluted sites bioremediation, this type of remediation. So if you have bacteria, you're augmenting it, it's called. Stimulating is with nutrients. Transform. Anyway, here we go. Stimulation is with nutrients. Like N and P. D. Professor Chakraborty, used to break down crude oil. Petita breaks down crude oil. Pseudomonas petita. If you want to culture, it doesn't matter. Identifying the major limiting factors by using microorganisms in developing bioremediation strategies. Mechanical. B. Word. Isolation and characterization of microorganisms. Environmental samples refer to extraction. Isolation. It's A, I reckon. A, yeah. Whole genome. I'm putting my hand on the screen so people are thinking, look, he's got the thing in front of him. I'm just putting my hand on the screen so I don't see the answers. Because I'm testing myself, so I'm trying to study, for this. And whole genome sequences of bacterium could be considered as part of a polyphasic taxonomy. Whole genome sequencing of bacteria. Yeah. During biotransformation of toxic organic compounds, they are oxidized by microbes, which result in the conversion of pollutant molecules to heaps of stuff. Because many microbes do many things, so to be. Which among the following is one of the most abundant and potent electron acceptors? Come on, I'll wait a bit. You know this. It's the stuff we breathe in. Oxygen's the most potent. Geobacter uraniandrescens uses uranium as electron donor. No, it's an electron acceptor. That's false. Dihalococcus species can bioremediate. It's your polychlorinated biphenyls by using it as a terminal on an electron acceptor in anaerobic conditions. Geobacter metalloregenesis produces flagella only when grown in presence of insoluble ferrous or manganese oxides. True. And remember the plant has a flagella gene, an FLG22 gene, which it can pick up. So if these are in presence of it with its flagella, the plant knows it and can act accordingly, putting out different terpenes. Which among the following contamination sources is not a point source, so which doesn't come from pharmaceuticals? Everything else comes from the point. Microbial cells interact with benzene and break it down to produce carbon dioxide and water. This is a not stimulating all that, or absorbing it, is degrading it. Microorganisms present in contaminated site use something to break down, or breaking down stuffs, enzymes. Organic compounds will be degraded by measurable extent only if the organism has appropriate breaking down enzymes again. Biodegradation of benzene by hydrocarbon degrading bacteria is a multi-enzymatic process. It sure is. For bioremediation of hexavalent chromium, which types of metal microbe interactions is most appropriate? You want to reduce it. Most metals just gets reduced. Yeah. Because it's usually in its toxic form, in its bad form, because it's been oxidized, so you want to reduce it and get it out of its toxic form. Among the following metals, which one is essential for microbe activities but execute toxic when present higher? This is an interesting, You got cadmium, copper, and mercury. It's amazing. It's the least you'd think. It's copper. In eukaryotic cells, low molecular weight binding proteins used to combat metal toxicity at low concentrations belong to the group known as Metallogens. A for apple. How much CNP ratio is generally sufficient to ensure unrestricted growth of microbes in aquifers? Well, it's 30 carbon, it's a 5 for nitrogen, and for phosphorus, well, you're going to have something, because that's the backbone of your nucleic DNA and stuff. So 1, 35, 1. Which among the following options is correct for successful utilization of hydrocarbon compounds by degrading bacterium at a contaminated environment? Which one of the following options is correct? Could have more. pH is the giveaway. Which of the following types of microbe metal interactions does not require metabolic energy? Well, you can absorb it. Just absorbs. That's from passive energy, passive uptake, not active. Something are the largest class of enzymes that catalyze most common reactions. Something of the largest class of enzymes. That's pretty obvious. Remember oxygen? Oxygen reductase. So there's heaps and heaps. It's needed the most. Biodegradation of chlorinated hydrocarbon compounds are achieved by reducing them. Whoops, sorry. Is a branch of environmental biotechnology that utilizes microorganisms. C. Microbes involved in bioleaching use ferric and metal sulfides as their no, accepted. They don't accept. They transform sourcing. Yes, these are as energy. 1 and 4. C. In which of the following processes is bioleaching metals are directly extracted from their ores without physical extraction? It's got to be on site. So on site is in situ. Good. Eutrophication leads to yes. Depletion, yes. Formation of forms, and yes. A. So excess chemicals, that's what it causes. Excess, if you have excess fertilizer, when you don't use it, it runs into streams and it does all this. So this is the whole purpose of changing things. During the biological phosphate removal process from wastewater, phosphate gets accumulated in the cells. It sure does. Enhanced biological phosphate removal, phosphate accumulation in organisms, and something of the key players. Glycogen. Accumulate back to produce. B. Key successful biological nitrogen removal is enriching nitric oxiding bacteria and inhibiting ammonia. That's false. If you inhibit ammonia, that means it's not going to fix nitrogen, so that can't be true. The Anamox bacteria processes something, wherein the Anamox process occurs in B. Microbial enhanced oil recovery is from Pseudomonas petita, and it is done with injection of the bacteria into the reservoir and promotion. C. Emerging pollutants are bad. Can cause. C. Monitoring. C. Among these, which is not the emerging pollutant: steroids, methane, hormones. Well, methane's been around for years. It's not getting more in the environment. It's not new. Yep. Among the following methods, this is the last one, by the way. Among the following methods, which are the biological methods removing emerging pollutants? Wetlands, they are so beneficial. 14.5% is removed through them, I think, through the terrestrial wetlands. Carbon fixation. Carbon sequestration. Identify the synthetic carbon dioxide fixation pathway. I know that one. Formulose. I'd say, I reckon. Yep. Flue or syngas can be fixed and utilised by microbes. Sure can. Among the following microbes, which is used to produce methane from solid waste? Definitely. Yes. No. D. It's D. Among the following bacteria which is used to generate electricity, you want your acidophiles to produce electricity. Acidophilum species. They generate millivolts, but the added millivolts adds up. Among the following ions, which one's highly soluble? It's A. Ferric, not ferrous. Among these, which compound was injected into that for a biomobilization of chromium? Chromium. What would it use for its energy source? Polylactate. Fertilizers used in bio-augmentation of Alaska's oil fill served as a something source of indigenous microbiota. A. Tick. All right. Done. Back to you guys. Is anyone watching? Holy shit, there's 8 people here. What the hell are you watching for? Thanks for watching. I shouldn't have said that. Jesus, that's so rude. Thank you for watching. That's why I put this show up. Amazing. All right. I've got a little bit of time. I can show you some slides from it. I'll say g'day for the people who are watching. How you going, Main Green, Banana Farmer? Brendan, White Ashes, Cool Hand Luke. Main Green, Vinny, Vin, Banana Farmer. American one, g'day, mate. You like the microbe stuff? This is all about microbes. Cool Hand Luke. Main Green, everyone, good on you, Main Green. Cool Hand Banana Farmer. Thanks. Is oxidizing the ammonia making it plant available? No. If you oxidize the ammonia, you need to nitrify it. So you want to take a nitrifying bacteria and it'll change it from NH3 to NH4, which is your ammonium, and then that will be into the cycle. That'll form its element. So it'll take it out of the gas, out of the air. I hope that helps. That was the question that I was just answering. Frutel just got home. Oh, good. That's it. No questions. Good. All right. I'll share some slides because I've got an hour to go till my exam, and I'm pretty up on my research on my things there. So I'll just go to here we go. This section. Oh, there's not too many. Good. So I'll just try and share my screen again, and I'll show you some of the stuff for this course. It's a really enjoyable course. I love micro courses. So this isn't all the slides for the whole of the course. This is just ones that I particularly liked out of them because there's hundreds and hundreds of slides. So I'll just show these few and see how we go. Superbug versus microbial resources. So they genetically engineer them to make them resistant to a lot of stuff and can handle really good stuff. Highly degradative, high bacterial survival. They make thicker cell walls. You can really play around with them. Activated sludge. Environmental. Retain slow growing biomass. Yeah, that's how to do it. More powerful analytic tools. These are good, the redox couplings, and they show you how things change from what into what. And when you go and add an electron acceptor, because oxygen is one of the best electron acceptors you can get, and it just transforms, and the energy required, actually there's probably a redox tower in here. Yeah, the death of the microbes, they mostly form into cell form, into their, not a cyst, where they into a, what's that thing called, where half of the prokaryotic cells has got the little cell at the back, the spore. Yeah, when they die, so they can regenerate. That's why they're everywhere. Proteomics. Where's the redox coupling? That's how to sort of understand a little bit how the processes of it goes. Here we go. This is how it kind of changes. There's glucose getting changed into carbon dioxide and then it getting changed into water with the adding of oxygen. These are all the different classes that you can get for your obligate and facultative. Obligate means you've got an obligation to do it, and facultative means you can sort of do both. You haven't got an obligation. Inox, yep. Scale. Oh, here we go. The redox tower. This gets a bit into it. You've got your EV, and it's your millivolts that it's measured in, and that's why you can use your acidophiles to produce this millivolt of electricity to make renewable electricity. Oh, that'll do. Fungi, yes. Fungi is a eukaryotic microorganism, multicellular. Size is between 5 and 20 micron, size of our trichome heads. Types of fungi. Yeah, there's only 3 major types. Multicellular filamentous, macroscopic filamentous, and single-celled microscopic yeasts. So the difference between yeast and mold is your yeast doesn't have all hyphae, like the hairy stuff that mold has. Yeast is the colourful ones. Well, it doesn't have the hyphae anyway. And their fruiting bodies is the mushrooms. Yeah, your oomocytes. Ooh, you don't want them. Pythium. Yuck. Well, what's the other bad ones? Fusarium. Yeah, those bad ones anyway. Bad cannabis, medicinal cannabis stuff. Mold, yeast, and mushrooms. Very good. Algae. Algae are floating eukaryotic microorganisms that assimilate energy from the light. Their sizes are 10 to 20 micron. Algae carry out oxygenic photosynthesis, making them being an autotroph. That's the difference between algae and cyanobacteria. Cyanobacteria are prokaryotic cells, which algae is eukaryotic, so they have different processes. So the algae will go and respire CO2, where the blue-green algae, being prokaryotic, will respire. Oh, hang on. Well, it's the opposite. Algae will respire oxygen. That's what it breathes out, and we will do the CO2. Jesus, sorry. Algae. Hope that comes through good. I don't want to get too confused. I'm trying to keep all those, like, my knowledge up for the exam. Algae should not be confused. Oh, look, algae should not be confused with prokaryotes. Oh, wow. First question, algae is based on chlorophyll and pigments. Okay. Oh, Jesus. Oh, okay. So that's really different regions. Your blue and red green algae, like your agar comes from, what was it again, Raptor? I can't remember. The brown, I think. Protozoas. Yeah, they're cool. They got amoebas in them. Look funky under that. There you go, amoeba. They change as they move, they morph. They're so cool. Flagellates, and that's FLG22 gene that the plant recognizes. Helminths are parasitic worms. Helminths, parasitic worms. So you don't want any of them. So people scratch their ass a fair bit, maybe you might have one of them in it. Elemental composition microbes. Yep, remember it was 18.5.2. And the average 50%. That was that was 2 of the questions, and just right there. I used to study culture medium. Maybe you can make peptic. These are simple culture medium broths. Yeah, just from your yeast and your beef extracts. Got a lot of goodness in it. Culture medium. All right, this gets a bit technical. I hope this doesn't go into it. Oh, it does. Geez. Chemical constitution, biomass, viabilities. Yeah, your dry weight of your cell is supposed to contain, same as humans, 70% water. Medium, all right. I've got to watch time too. It's going all right. maybe another 10 or 15 minutes of this, and that'll be it. Exospore. Oh, here we go. Endospores. That was from bacteria, prokaryotic cells. They make a spore, which I just couldn't think of before. And half of it is the spore, but the back half of it. So see here, it's like a long looking tic tac, and half of it, the back, is the spore. So when it dies off, it can reproduce itself. And the endo's in, and exo's on the outside when it forms on the outside. Like see, here's the outs, and this one's the in. Cool. S phase. I did cell biology just to learn this about prokaryotic cell cycle. Reproduces from budding fission. Exponential microbe growth and equations. Oh, yeah. So the growth and yield calculations. Yes. Oops, quick. Effective environment and growth in microbes. Yeah, you need all those things for them to grow successfully. Effective nutrients, it's your formulation. Effectiveness of pHs, yes. Acidophiles, neurophiles, alkophiles. So neutrophile is growth mediums referring that. Acidophiles below 5 and alkos over 8. Righto. Carbon energy sources. Glucose does both. Wow. Two types of questions here. First, what drives metabolism? Energy. Oh, what are the energy and carbon sources? Okay. This is how to get into the full-on terminology of it. Try and pronounce some of them. Chemosynthetic lithoheterotrophs. They use inorganic compounds. You can, like chemo is chemical, and your synthetic is your man-made type stuff. Lithotrophs, rocks. Hetero means it's not similar, so it'll be multiple things. So you can, photo means from the sun or light. So you can kind of break it down from just the Latin words that are inside of it. Oh, here's your catabolism, where it makes the energy. No, anabolism makes it, and catabolism uses it. Energy source comes in. What did they ask about? Because look, your carbon source is coming in, and it's making it here, and you're catabolizing it, and then you're anabolizing it, and it's processing it. Biosynthesis, polymerizing, macromerizing. I think I've got that asked about. Shit it. All right. Oh, well, it's not for now. General trends in the proportions of genes related to the 4 prokaryotic stages. Rarity is extended periods of rapid. Yeah, look at this. This is a rad one. In 48 hours, a single bacterium, just a bacteria. You know, the lifespan of a bacteria is a dot on a piece of paper. So in 48 hours, one single bacteria can exponentially double its biomass every 20 minutes. If it was in perfect conditions, it would roughly cover 4,000 times the weight of the Earth in 2 days. Yeah, it's fascinating. That just shows you that microbes, they need the right conditions to reproduce in. And once they get too favourable, or there's too much of this, or they've expired too much of that, they'll stop their growth, and they'll even go into spore form. Biogeochemical mechanism ATP production relies on. That's pretty technical. This is some cell coupling. This is cool. This is like the cell wall. All right, this goes into detail. All right, it's really detailed. Free energy. Metabolism. 8 common processes that are recognized to occur in carbon-rich habitats. Denitric iron fermentation. Oh, this is how it goes. This is the process from, it's good, the energy. Oh, right. So see up here, from aerobic respiration with your oxygen as an electron acceptor, you can produce the microbes can get up to 13 millivolts of energy requiring here, and negative 25 kilojoules equivalent that they can get out of it. As it goes down, they get less and less and less, but they'll need something to get their energy from. So they'll still, if oxygen's gone, they'll go with nitrate. Then they'll go down with manganese, and then they'll go down with, if it's available, iron, sulfate, and just etc. etc. It's pretty cool. So that's how it processes. So you can tell from which stage what you got growing to what's there and what's happening by just doing a quick analysis of this labs. This is that's what I was just talking about then. When they lose their electrons, they'll go into the next stage, and then they'll need an electron acceptor, so they'll go into the next stage again. They'll keep going down. That's its rate that it goes down. Electron accepting process. That characterizes downgrading. Oh yeah, so it's if you have an oil spill, it just shows you how it depletes over time. So that's your timeline, and then it should go from your methane to your hydrogen sulfate to your iron to manganese, and then slowly get into your oxygen. So they'll grab some nitrogen sources, and then it'll be sort of fixed. So that's how biology naturally does it. Catabolism, network of reactions, ATP. For generation, I did have it asked about. It's the so catabolism makes it, and anabolism uses it. Oxidizes, yep, makes it, yep, and then anabolism uses it. Okay, so cats first. Big cat equipment. I remember it like that. Biochemical manifestation of catabolism. This goes into a little bit of detail. A hell of a lot of detail. Look, 6 ATP. Yeah, produce ATP from glycolysis. Anyway, this is a redox reducing scale. It sort of breaks everything down. It shows you can take nitrate. Nitrate is reduced to dinitrogen, and then same thing here, and then it goes down and down. That's where you get your energy gain. There's less and less, like I said before. And this is your amount of millivolts that you'll get measured in EO and your EH. And that's a pH scale. Measured in PE. No, it should be anyway. Syntropy. All right. And really interspecies hydrogen. Following a spill. That's how it works. Rate limiting steps. Single cell identification in microbial communities. Yes. We want to amplify it. qPCR. Preparation of soil. That's how you do most probable, no, not the most probable number, your great plate count anomaly. Serial dilution is the word I was looking for. Functional genes. Yes, these are cool gene names that you want to remember. It's fun. See the genes, they're mostly related to what the process is. Like nitrate is your NIR. So NIR, nitrite is your NAR is nitrate, and NIR is nitrite. Actually, this should be nitrite. They've written that wrong. This is nitrite. That's actually what it is. Your NAs is nitrate, and NI is your nitrite. So it's just a way to remember it. They're very similar, the genes. Like your early finishing gene in plants is the ELF gene. 6 categories to culturability. Okay, microbial, broad outlines, single microbial methods, direct isolation, select conditions, select enrichment culture. Enrichment cultures are rad. See this, you can make, actually, they're not talking about that. Quantification of, I got to watch with YouTube because I'm getting strikes and stuff. So I got to try and talk about legal things and promote legal stuff and safe use of things, which I really, to be honest, I do anyway, because it's there. I've got a workplace health and safety background, and that's what I really truly believe in. It's the only way to do it safely, and for the environment too. Isolation of anaerobic bacteria. Yeah, you don't want any oxygen for that stuff. Most probable number, MPN, with your serial dilution. Serial dilution, the inoculant, used successfully to isolate. That's how you can see it's pretty. Alternative methods for isolation of pure microbial methods. Culture purity. The purity is isolated culture is made through the combination of microscopy, colony characteristics, and testing for the cultures on growth mediums. Identification. List of prokaryotic strains from the nomenclature. Nomenclature means what the actual terminology means. That's the gospel. The dictionary. Process-centric. This is to enrich halate. I'll have to skip through that stuff. This is some Pseudomonas strain data, DNA data. Summary of culture-independent methods. Staining methods. DAPL. Yep, binds it's fluorescent though. There it is, fluoro. I don't have a fluorescent. I got polymerized lenses for my microscope, and I'm still yet to play around with this. Viability staining. Yes, 2 dyes are used. Based on that's to see which ones are dead or alive, which ones penetrate the cell or not. Yeah, your CYP09 dye penetrates all membranes, stains all the cells, but PI only penetrates damaged cells, meaning the dead ones. So that's an easy way to see the viability. Limitations. Yeah, you can only go down to 100 nanometers. Look at this. This is a picture that they did with microbes. So, and then they stained the various types, and it came out with this beautiful color, this beautiful picture. So it's a microbe painting. GFPs. Righto, I think we're finished here. That's it. Can't believe people are watching. Thanks for that. Well, I'm just going to scroll up a bit. Microbes are rad, says Fratel. They sure are. Crueland Luke. I thought the mulching module was next. Crueland, that's on next weekend, mate. It sure is. Going about mulching. These are just a study session for my exams. This reminds me of a free game. You can float around the cells. Oh, Crueland Luke, that's the way. Sunshine, g'day, mate. Is philosopher's stone still legal? I don't know. I promote legal stuff though. Kata meaning change. Don't know what that means. When I was a diesel mechanic, we had a diesel bug in the underground tank that fed the Bowser. It was a nightmare. Yeah, mate. That's right. Very good. Haha. CC correcting. No, it's not the professors. It's a lot of these things are written by their teachers' aides and the student aides, and they skim through them, and then they'll give the stuff for other people to write. So quite a lot of the classes, the professors end up correcting the terminology. That's not right, or they'll write on the screen and stuff like that. So it's yeah, there is some errors sometimes. They're not really fact checked as well as they could. But, oh well. Good on you, American One. Interesting stuff. Thanks for sharing. Thanks, mate. This is my session's going to end. I've got my exam in 43 minutes for this. I'm going to do another one of these this afternoon, and really, truly, it's for entrepreneurship. It will be boring as hell. I shouldn't say that, but it's really interesting for the people who like it. But for the plant soil science community, it's probably not going to be that interesting. It's more interesting for the business type or the persons that want to not work for other people. And that's really what the course attains to, but I'll be going through the questions. And those questions are more common sense, business-like questions on how to set up and idealization and how it runs and that sort of stuff. So, yeah, that's happening in a few hours after I do this exam. So I'll get back to studying, and then the next one's coming up. So thanks, everybody, for writing some stuff in and saying g'day and that sort of stuff. Yeah. I appreciate everybody's effort, making the effort. You didn't have to. You could probably be doing other stuff at the moment. So, thank you. Thanks again. White ashes. Wow, I want to make a micro painting. Yeah, you have to do it with stains. There's a few simple stains that you can get to do it with, and gosh, yeah. Then you got to make sure that it doesn't contaminate and stuff too. So it might be set up your little tissue culture lab first, and then it'll happen really good. You can make one of them out of a tote. Or a big, one of those, what are Australians called totes? The big plastic-looking box things. And you make one out of that, turn it on its side, and you put a little flap over the top, and an exit fan, and a Bunsen burner, and UVC light, and that's a pretty cool, cheap alternative. Set up your tissue culture lab. So you can do that in that, play around with microbes. Cool, hand Luke. I thought it was sick too, Ashes. Just want to scope your worm castings, mate. Yeah, banana farmer. You don't need good luck, so I'll say good memories. Good on you, American one. Yeah, always need good luck, because it's always, they throw in just heaps of random questions, and my English isn't the best, so I falter in some areas. But appreciate your kind words, everybody. And thank you again. And I hope you all have a really nice day. I'll see you next time. I'll be doing this again in a couple of hours just for another study session. And next Saturday will be the normal plant soil science and microbiology on medical cannabis related stuff. Cheers, everybody. Happy breeding, happy growing, and good health to you all. Thank you. Bye-bye.